have the pleasure of introducing Angie Seebach from Williams Brothers Pharmacy, and today she's going to be talking about pharmacogenomics.
Good job.
Yeah, I practiced just a little bit, apparently not quite enough. But Angie, thank you and welcome.
Thank you very much. So I am a consultant pharmacist.
Can you talk really loudly?
Yes, thank you. And tell me if you can't hear me because I do get soft. So I am a consultant pharmacist. I have been for 14 years with Williams Brothers Pharmacy. And I did a certificate course on pharmacogenomics several years ago when this was just starting to come out. Oh, does that help?
Yeah.
All right. Okay, so I'll stay away from everybody while I'm up here without my mask. But also, there's also a new drug called Aduhelm that was recently FDA approved over the summer. So once we get through this, if we have a little bit of time at the end, I'll just kind of go over that because a lot of people have questions about that and are interested in learning about that as well, since I'm a pharmacist and that's kind of my area. So we'll start with the pharmacogenomics and how it can help personalize medication. So I'll describe what pharmacogenomics is and kind of give you the basic science behind it. I'll try not to go too far into it and bore everyone to death today. But I'll also try and tell you kind of how it's used and why it's useful in medication therapy. So first off, a lot of people interchange pharmacogenomics and pharmacogenetics. You'll hear that both of those terms. Pharmacogenetics basically just refers to how one gene variant affects medications, as pharmacogenomics refers to how multiple genes up to the whole genome can affect medications. So people respond differently to medications. Most respond well, and their health improves, and everything's great. Some people tend to gain no benefit, or they think maybe their drugs aren't working, or certain drugs aren't working for them. And then other people maybe have side effects, or feel like something feels a little too strong for them, or has an adverse reaction to that medication. And this is because after you take a medication, it gets metabolized or processed by your body. And how it is processed by your body is determined by how your genes work and what your genes are telling it to do. So we'll start with Genetics 101. Humans share about 99.5% of their genomes. It's that other 0.5% that's different for everyone that can affect how susceptible we are to medications and how we respond to different drugs. So we have about 20,000 genes that make proteins in our body. And the proteins are what make us unique. They determine hair color, eye color, but they also determine how we respond to medications. So we think of DNA as a code of letters, and A, C, G, and T are the letters we typically use to spell out different instructions that we call genes. So the genes tell our cells how to make proteins, and then sometimes in these proteins we'll have a letter replaced, like an A with a T, that isn't usually there. And that kind of tells the protein to give different instructions to our cells. And it can change how the protein works and how we metabolize medications. And then this is as far as we'll go before I bore everyone to death, but we'll talk a little bit about alleles. So alleles are the different forms of genes found on a specific chromosome. So everyone has two chromosomes. We have the mom chromosome and the dad chromosome. And on each chromosome, we have an allele in a specific position of the DNA. The most common types of alleles are called wild-type alleles. That's just what we consider like our normal alleles. But there are also variant alleles, or some people refer to them as mutations that are less common. And these are the ones that cause like things like different responses to medications. So you'll also see terms like homozygous and heterozygous. So if both chromosomes, the mom and dad chromosome, both have the same, whether it's the wild type or the same variant, then it's homozygous. But sometimes they can have maybe a wild type and a variant, which makes it heterozygous. It makes it a little bit more cloudy as to what's going on. So these genetic variations can alter things like how much of a medication gets absorbed, how well it gets to where it needs to go, how well a medication gets broken down in the body. And they can also determine how your body responds to medications if it thinks it's something foreign and you have an allergic reaction to that medication. So why is pharmacogenomics important? How are we going to use this information? It helps us first identify a person who might be more or less likely to respond to a drug. Maybe if we need to start with a higher or lower dose of a drug, or possibly if we might just need to avoid a drug altogether. So for say if they might have an allergic reaction, like with carbamazepines, ones I think of, there is a specific population that the FDA labeling recommends having genetic variation or genetic testing done for, because they are more likely to have Stevens-Johnson syndrome. So they want you to test for what's called the HLA-B 1502 allele. And that sometimes can determine if someone's more likely to have that severe allergic reaction. And if they do, we want to use a different medication because we don't want them to have that. So we have different types of metabolizers that determine how we're going to react to a medication, or how we're going to process a medication. We have our ultra-rapid metabolizers that are going to go through these drugs pretty fast. Then it's always been called extensive metabolizers, or we also sometimes refer to them as normal metabolizers because extensive seems fast, but really they're just metabolizing drugs normally as we would expect. We have intermediate metabolizers that kind of process drugs a little bit more slowly, but not quite as slowly as like a poor metabolizer who might not even process a drug at all. And then, to make it even more confusing, we have prodrugs and active drugs. So we're going to start with an active drug. This is just a normal drug. You take the drug, your body breaks it down to an inactive form, and you get rid of it. So when you take it in active form, here is 6-mercaptopurine as our example. We have a normal metabolizer at the top, so they need a certain amount of the drug, represented by the pills over there. They go through it, metabolize it normally, it works fine. But then we have some people that are the green guys. These aren't very clear colors, but or the maybe yellowish guys, that they are maybe intermediate metabolizers. They aren't going to process it as fast. So the drug can kind of build up in their system while it's waiting to get changed to an inactive drug and eliminated. So for those, we might need a little less drug, so it's not building up and causing toxicity. But then here at the bottom, we have the little pink guy. He is a poor metabolizer, and he can't process the drug. It just builds up in his system, causes toxicity, and with this particular drug, it can actually be fatal. So with our poor metabolizers, depending on the patient and the drug, we might use a really low dose of the drug if it's something that we really need to use, or if it's something as severe as possibly causing a fatality, then we would try to use another drug if at all possible compared to that one. Oops. I'm not good with flipper today. One more time. Okay. So at the bottom, that example is shown in a different way. Again, we have normal function, we get the expected effect. The decreased function, or our intermediate metabolizer, risk of hematologic toxicity, and then if we have a poor metabolizer, a greater risk of toxicity. With a prodrug, we give the drug in an inactive form because maybe the active form would get degraded in the stomach as it's getting, metabolized and going through our system. So we have to give it in an inactive form, then it is metabolized to an active form that does what it's supposed to do in the body. So with these drugs, if we have increased function or the ultra-rapid metabolizers, then we're going to be turning this drug into the active form a lot faster. So it's going to have a higher effect, more toxicity, and we want to use lower doses possibly in these patients. So one example that we have up here is codeine. Codeine gets transformed into morphine in the body after it gets metabolized, and then the morphine is the one that actually works for pain. And so on the other side, if we have somebody with decreased or no function, since codeine is an inactive drug and we're not turning it into the active drug anymore because we can't metabolize it, then they're going to feel like it doesn't really do anything for their pain and it's not really working for them. So the people that are on like Tylenol number 3 or Tramadol is another active, or prodrug, sorry, these patients might say, I feel like it's not really working, it's not really relieving the pain. And that's because it could possibly be because it's not being converted to the active form. And it could be a genetic thing where if we did genetic testing, we could see that, you know, that gene is not working very well for them and we need to try a different medication. So I just put up a few more common drugs that we use in long-term care that have FDA labels. So codeine, tramadol, Celebrex are the pain ones. Some cardiovascular ones we see with pharmacogenomic labeling would be warfarin and Plavix or clopidogrel. And then some of the psychoactive ones, citalopram, fluoxetine, some of the antipsychotics, several of the antidepressants have pharmacogenomic information. And this is definitely not all-inclusive. There are about, I think it was 715 drugs the last time I checked the website that have pharmacogenomic information associated with them. So here's just an example of what the FDA labeling looks like. If you are a nurse, doctor, pharmacist, you know, you might see this at some point if you look for it. Otherwise, it might mean absolutely nothing to you, so I'll go over it really quickly. But there's a couple different genes that, a couple different enzymes that break down coumadin. And on the left side is our normal people that we usually start out, or normal pills that we usually start out at about 5 milligrams starting dose of warfarin. But then as you go further to the right, you'll see the star 3, which means these are lower functioning alleles. And on those patients, they're not going to be able to process it fast enough, so we have an increased risk of bleeding with those patients compared to somebody who's a normal metabolizer. So for those patients, we might start out at 0.5 to 2 milligrams. So this is actually in warfarin's package insert. So how many people have done pharmacogenomic testing for warfarin? Anyone? So we don't see this. It's still really, really new, and we haven't really gotten around to doing this a lot. So the information is there, and they're doing more and more studies and finding more and more out about this, but a lot of people still don't know about it. It's not something that's common practice yet. So pharmacogenomic testing is something that we can do for a single drug gene pair. It is, we can also do it for multiple genes, and it gives us an insight into how an individual is going to respond to a medication, possibly. And then we can use that and kind of tailor their treatment to that individual. Maybe give them a higher or lower starting dose, or maybe pick a different medication if it doesn't look like that one will work at all. So the test can be collected via blood sample, saliva, or the most common is a cheek swab. It just looks like a really big long Q-tip that you swab your cheek for 10 seconds, and then you'll stick it in a vial and send it off to a lab. Turnaround times vary. We don't see a lot of on-site labs yet because it is expensive and it's not very common yet. So we don't see a lot of in-house labs. We see more often labs where we have to send it out and then we get the results back maybe within a few weeks. Sometimes it's sooner, sometimes it's later. It just depends on what lab you're sending it to. And then the tests need to be ordered by a practitioner, usually their physician, and it has to be for a specific reason. So the most common ones I see are like a cardiovascular panel, or a psychoactive panel, or a pain panel. Those are the ones that we use most commonly in long-term care to help us pick the correct drug. Oops. Okay. So there are some challenges to pharmacogenomics. Like many of you, I saw no hands come up when I asked if we did any testing for warfarin. It's not common. We haven't started doing it a lot. It's likely to become a very big part of future care, but right now it's still being studied. We have a lot to learn before we really start using this widespread. It's rare for the drug's response to be affected by pharmacogenomics. So, I didn't put the picture in here, but there's, like we said, it's most likely people will respond normally to a medication. It's just the outliers that we're looking for. So, since it isn't everybody, it's not something that we're in a hurry to do yet. Also, there are a lot of interacting medications that could also be the cause. So maybe there's a drug interaction along with the pharmacogenomics on top of something else causing different responses to medication. So it's not just the pharmacogenomics. So it's pretty complex, and they're trying to figure out like how it fits in with everything else still. And while a genetic variant can increase the likelihood that a drug might be metabolized or react a certain way, it doesn't guarantee it will. So like I said, with the carbamazepine and the Stevens-Johnson syndrome, it's more likely that that population will have that reaction, but it doesn't guarantee that they will have it. And not having that gene also doesn't guarantee that they won't have it. There could be another gene that we don't know about yet that's causing that as well. And so even when we find a correlation between a genetic variation and a medication response, then we have to develop a test. We have to prove that it's beneficial in trials. We also have to prove that it's cost effective. So if it's, something just gets metabolized a little bit more slowly, but you're really, it's not a big drug, you don't really have to worry about changing the dose that much in the patient, then what's the point in spending the money on the test if it's not going to change health practice? So that's something else to keep in mind. The testing can be costly. It can run between $200 and $500, although there is some assistance, and some labs will have an amazing sliding scale payment. And there's an increasing trend of insurance coverage, but it's still not covered widely. So most of the time people are paying out of pocket, and if it's expensive and they don't know if it's going to do anything, they don't really want to pay for it, and I don't blame them. Yep. Is that the last one? Thanks. So I put a couple of helpful resources. My favorite one is the Pharmacogenetics. It kind of gives an overview of pharmacogenomics, what it is, how it affects medications, and also, some of the medications and some of the alleles that we're looking at. Also, there's the Clinical Pharmacogenetics Implementation Consortium. This one is more drug specific, and it looks at particular drugs and what the alleles are that we're looking for each of those drugs, if you're ever looking for like a specific medication or a specific genetic variant. So I will stop there with that. Do you guys have any questions?
Angie, does Medicare pay for this?
No, not yet. At least not that I know of.
You were talking about how some people absorb or, what are the two categories, but
Metabolites.
yeah, and some people do and some people don't. And are there, like, is it like some people are just this way and some people are just that way? Or is it various degrees in between?
Between the different metabolizations. So that's where it gets a little cloudy because, like with warfarin, we saw that it had the B4C1 and also had the CYP2C19, I think. And so there's two different ways that it's metabolized. So if maybe one is a poor metabolizer and the other one is a normal metabolizer, does that make them an intermediate metabolizer? Maybe. But maybe the normal metabolization is the pathway that it goes through more so than the poor metabolization pathway. So maybe they're like leaning a little bit more towards being a normal metabolizer. So that's where, it takes somebody who's really studied this and really knows the difference between all the different pathways and, what percent of a drug gets metabolized this way versus this way to figure out, is this going to affect the dose that we need, and is this going to make any difference in how we treat the patient? So, does that answer? Any other questions?
Are there specific drugs that we know are being used for dementia-related diseases and disorders that are more likely to be affected by metabolism? Does that make sense?
Yeah, so there are some antidepressants and there are some antipsychotics that are metabolized, and different ones are metabolized by different enzymes. So that's why sometimes we get a psychoactive panel. But specifically, dementia medications like your Aricept and Namenda, they don't really have a lot of information on those yet that I know of. but sometimes, like, you start somebody on an antidepressant and it doesn't seem to work as well, so you switch them to another one and then it works. That could possibly be because of the way they're metabolizing it, and because of the enzymes and the genetic variation.
If you have that testing done, does it test for one medication at a time? Or does it test, how does that work?
So typically we'll test for the allele. So our CYP enzymes are a big way that our drugs are metabolized. So maybe we'll test for like this particular CYP2D6. But a lot of drugs get metabolized through that one pathway. So they'll look at the allele, and then a lot of the printouts that I will see will say, okay, because of this type of metabolization of this allele, we have this set of drugs that might be affected, you know, either increase or decrease the dose, that kind of thing.
Could you play devil's advocate for a moment and address the issue of, a friend of mine wrote a book called Drug-Induced Dementia, where medications are actually inducing various dementias, and the studies that they've done, and just comment about that.
So yes, drugs can have a lot of adverse effects. So a lot of them can cause different cognitive reactions, and even like some of our drugs can cause Parkinson-like reactions. There are different adverse reactions of medications. So I don't know specifically if that's something that we are able to check for yet with pharmacogenomics. I haven't seen anything specifically on that with pharmacogenomics. But that could be something in the future that we might find. Again, it's still such a growing field. There's so many things that we're finding out that we don't know.
Thank you.
Any other questions?
I have another one.
Sure.
So there's such popularity with at-home genetic testing now.
Mm-hmm.
Do you think that that could be something we see in the future with like 23andMe offers these allele testing as well?
So 23andMe, actually, a long time ago, this was from my genetics course that I took like 5 years ago. So, but even before that, they would actually give you like, you know, what kind of alleles you have and like different medication things. They didn't have all the information back then. So there weren't as many things to report. They don't give you that anymore. From what I've heard, it was partially because people were kind of like freaking out about all this stuff that they have. But back then, I don't know if this is still true, you were able to actually still download your entire genome.
Yeah.
Wow.
And then you can search for a specific SNP is what we call them, or
Because
specific
they're
allele.
so affordable, especially they'll do like a Black Friday sale. It'll probably be $100. Like if people could do that.
Yeah. So yeah, because back then, and then you could search for a specific allele if you wanted to know if you had like there's like the MTHFR gene had different alleles that can make you more susceptible to like cardiovascular disease. And so you can find that in there.
Via Dr. Rudy Tanzi, who was in our film, he has told me, because I asked him about this issue, because this is a statistic that's growing. Dr. Tanzi in our film, I had asked him this very same question because I've noticed statistics after post-surgery with symptoms of dementia, but they don't leave either. It's not like they came and then they left. So it was a process of dementia developed after surgery within literally a few months of the surgery. Mm-hmm. And he said, God, I hope I'm getting this exactly right, because I usually refer to notes when I give this information. Isoflurane is one of the things that they put in a formula for anesthesia. They also, even when you get a colonoscopy, will give you something called an amnesiac. So when I have had, although now I take colonoscopies without any anesthesia because I become very cautious, but I always tell them, do not put the amnesiac in my, when I used to say it, don't put the amnesiac in. I don't care if I remember, oh God, that did hurt, or whatever I heard, it's irrelevant. But these are two chemicals that I want you to remember and write them down, because it's isoflurane is, and this is why I wish I had my notes with me, because I had them prepared earlier and then I put them in the car. So Isoflurane, isoflurane, is one of the substances that are placed in an anesthesia typically. I said, what would you suggest for your mom if she were going into surgery? Because I figured he's more protective of his family. And he said, I would have, and this is what I want to make sure that, God, I would feel horrible professionally if I make this mistake. So check me. Isoflurane should be replaced. And I will go to the car during intermission and run up at the microphone and announce this to correct myself if I'm wrong. With desiflurane. And the D-E-S-Y, I think, again, I got it spelled correctly in my notes. D-E-S-Y-F-L-U-R-A-N-E. And so that you would prefer, and then I'm going to give you the correction after lunch. I mean, after the workshops. I'll go check it. But one is a much more favorable for not having dementia via the anesthesia. So that's all what I could say about those two chemicals in anesthesia, but I don't want to make a mistake and tell you which one is the good one and which one is not. Isoflurane and desiflurane.
That's very interesting, yes.
Thank
So I don't know a lot of surgery drugs being involved in parasites.
Yeah, and that's through a professional, not me telling you my opinion.
Thank you.
But it's a fact of surgery.
Yeah, thank you.
I think this has been commonly asked, but do you have any new information about things like 23andMe or what's the other one with the
Ancestry.
With any of those, is there still protection from MIB, from the Medical Information Bureau, gaining that information that can affect your insurability and so forth?
That is a very good question. 'Cause I know that's something that everybody's always worried about, concerned about. And these companies have been trying to protect your information as much as possible, especially, the ones that are doing the pharmacogenomic testing. But again, we never know. I mean, there are HIPAA regulations and all that, but that is a good question.
My understanding is that they are still protecting your information, but there's if you're interested in sharing your genetic information for other reasons, there's a separate independent website where you can upload it yourself. Only know that because of true crime podcasts. But
No, I mean, I know they've always been in the past. It's always a fluid thing, though, and you would hope that they would never get to a point where they would share it. But yes, that is something that everybody's always been concerned about.
I would hope or would like to see a company or a site that offers that, but they destroy your information after sending it to you, and that there's no retention of that information.
That's a great idea.
Yeah, because that's what I hate the thought of, is that information staying out there, just like anything you say on the internet is there forever.
Yes.
And I hate the thought of that information maybe safe for 5 or 10 years, and then all of a sudden some legislation comes down and boom, it's all in government's hands or whatever.
I mean, and from a medical standpoint, it should be protected by the HIPAA regulations. And when your doctors have it, it's good that they hang on to it, because a new gene might come up and then they can, go through the results again if they have your old genome and see if you have that gene and if it could affect your medications or, put you at risk for a certain disease state or something in the future.
I do have my notes. I was correct, but I want to make sure. Isoflurane, and substitute isoflurane with desflurane, D-E-S-F-L-U-R-A-N-E. He said that would be a preferable substitution on that medication, anesthetic. Thank you.
Any other questions? Do you guys want to talk about Aduhelm? Has anybody heard of this medication yet?
Yes.
Okay. And it's aducanumab, in case anybody wants to know how to pronounce it. There we go. Okay, so this was approved in June of this year. It is approved for mild cognitive impairment related to Alzheimer's disease, as well as mild Alzheimer's disease. It is an IV infusion, so it is an injection. You have to give it over an hour and 100 mils of normal saline, and you have to give it every 4 weeks. So it's not the easiest administration for a medication. It starts with weight-based dosing, and then you titrate it up over the first 7 administrations, so basically the first 7 months of doing this medication. And depending on your dose, the average is about $56,000 a year. So I doubt any insurance has picked this up yet because it's so new, but maybe, I don't know. It's kind of a controversial drug, so they might wait a little bit longer than usual to pick it up. But just, again.
It won't be approved officially until 2030. That's a conditional approval.
Yes. Good job. You did your research. So a quick background on Alzheimer's disease. So in studies, we found a couple of different ways that we think Alzheimer's disease is caused. One is there's a buildup of these beta amyloid plaques outside of your neurons that inhibits the communication from one neuron to the next. And there's also these things called tau tangles that build up inside the neurons and eventually end up causing nerve death. So Aduhelm was approved based on the reduction of these beta amyloid plaques in the brain. But I will say that it's not just the plaques and tangles. There have been studies where people have had dementia and have had no disease evidence in their brain whatsoever from these. So it's still an ongoing thing. There's still many variables to Alzheimer's disease that we don't know, but these are the 2 that we know. So like Aricept and Exelon and what's galantamine is the other one. Those all attack the amyloid plaques, and the tau tangles, the only drug so far that we know of for that one is Namenda. So the timeline to approval. In October of 2018, they saw some possible promising results in their phase 1b trials. So they said, okay, let's make this a larger trial and see if we can get this drug approved. But in March of 2019, they ended the phase 3 trials early because they saw no benefit. Then they came back in October 2019 and said, wait, we did the sub-analysis of these groups and we want regulatory approval because we think we might have some benefit here. And their sub-analysis, so there are 2 different groups in the ENGAGE study. One was a low-dose group and the other one was a high-dose group. And so in the high-dose group, that's the group that they saw there might be some possible benefit in. So July 2020, they submitted the FDA application, and then that was June 2021. The FDA granted accelerated approval, even though the advisory committee that usually gives the recommendation to the FDA said almost unanimously that no, we don't approve this. We don't think it's had enough efficacy studies to prove that it's going to be effective. So then the FDA, I think it was the next month actually, they said, well, continued approval is going to be contingent upon verification of the clinical benefits in ongoing trials. So the trials that they were talking about back then are supposed to wrap up in September of 2023. So we're still doing trials. There might be some benefit, but it's kind of iffy. We don't really know for sure how much benefit there is just yet. I cannot use a clicker today, apparently. There we go. So there are a few adverse effects, but the most prominent one that I want to talk about is called ARIA, or amyloid-related imaging abnormalities. It was seen in the trials, and it most commonly presents as a temporary swelling in the brain that resolves after a while. But there's also a possibility there might be some small spots of bleeding, and that's either on the surface or close to the surface of the brain. Those are called microhemorrhages. And sometimes if there's enough of these microhemorrhages, it can cause blood to build up in the brain and can cause seizures. Usually there are no symptoms though, at first at least. Maybe some dizziness, confusion, headaches, or vision changes or nausea. But typically patients will see no side effects. So that's why they recommend monitoring when you start Aduhelm. First, you want a PET scan sometime within the first year of treatment. But you also want to make sure to get an MRI prior to treatment, prior to the 7th dose, which will be about 6 months in, and prior to the 12th dose, which would be 11 months in. If it's observed, they recommend stopping the therapy and having the prescriber evaluate risk versus benefits. And contraindications, there are none that are listed in the package labeling. But when they did the trials, they excluded a lot of patients. They excluded patients with stroke or TIA, any type of dementia other than Alzheimer's dementia, because it's only approved for Alzheimer's dementia specifically. So if somebody has vascular dementia or Lewy body dementia, they haven't approved this for those indications. Myocardial infarction or heart attacks. Also, they excluded a lot more people, like anybody with liver or renal function. Well, most of our elderly patients are going to have some renal impairment, or renal function impairment, sorry. Anybody with brain hemorrhage or using blood thinners, so a lot of our patients are going to be on some aspirin or some warfarin or something like that. So I would say if somebody has one of these indications, I would be cautious about starting it just because we haven't tried it in any of these patients yet. So in summary, it's a first-in-class drug, which means there is no other drug just like it out on the market yet. It is directed at those beta amyloid plaques. It is a monoclonal antibody. It is approved for earlier stages of dementia compared to what we have approved now currently. It will help, or it is thought to help to slow the progression of dementia, but we still don't have any medication that is going to reverse or stop dementia completely. It is an IV infusion, so again, there's a challenge of actually giving the medication. It has to be given IV every 4 weeks. It's given indefinitely. There is something in the package insert that says you may be able to spread out the infusions over longer than 4 weeks if they start to see a reduction of plaques, but there is no timeline listed. They need to finish their ongoing studies before they know for sure what that should be. So most common adverse reactions we talked about: ARIA, there's also headache, and so falls, of course, which is something that we definitely want to watch for in our elderly patients. And we have limited studies on the efficacy and the long-term safety of this medication, and it's pretty expensive. So those are just some of the thoughts, and if anybody is interested in learning about the new drug. Does anybody have any questions?
Have you seen anyone have that prescribed yet that you've filled? No.
No, I haven't yet. No, not yet. Not yet. It's pretty new. I think a lot of people are being cautious right now, kind of waiting for the rest of the trials to wrap up.
And not dropping 60 grand on something like this.
Yes, we have a lot less expensive. I mean, they're still expensive, but a lot less expensive medications out there right now. Well, thank you guys for your time.